Thank you for joining this webinar series about CMT. Today is the last episode of the CMT webinar series co-organized by the European Academy of Neurology and EURO-NMD for rare neurological diseases. And this webinar series was held under the auspice of the European CMT Research Association, ECRA. Please note that today's webinar is recorded and made available on our website and YouTube channel. So today we are happy to have with us Professor Kleopas, which is a senior consultant neurologist at the Cyprus Institute of Neurology and Genetics. He is also our co-chair of the peripheral nerve diseases and also the co-chair of the gene therapy of the European Reference Network for ERN, EURO-NMD, R&D, and EpiCare. Thank you, Kleopas, for being with us today, and the floor is totally yours.
Yeah, thank you, Elizabeth. Thank you for organizing this webinar series, and thank you all for joining. So this last presentation on Charcot Marie Tooth neuropathies will be focusing on current progress in treatments. So before going into treatments, just a few introductory lines about the Charcot Marie Tooth neuropathies. These are heterogeneous disorders with estimated prevalence altogether of 1 in 2,500 to 1 to 3,500 worldwide, but each subtype can be quite rare and many times ultra rare. There is a great phenotypic variability, including the age of onset and severity, and the disease may start from childhood all the way to old age. The presentation is characteristic with weakness and atrophy of distal muscles, difficulty walking, impaired balance, sensory loss, and there is a slow progression and gradually increasing disability as illustrated in these images from David's review. We have a great genetic and biological heterogeneity underlying this clinical variability. We have dominant, recessive, and X-linked forms. We have more than 130 causative genes, and we have demyelinating axonal and intermediate types. And this is classified based on the degree of slowing of the conduction velocity, but also reflects the biological basis of each subtype. So if we look at the molecular genetics of CMT neuropathies as a starting point for discussing therapies, we can recognize that we have many different cellular functions of CMT genes, and therefore many different mechanisms that lead to disease pathology and manifestations. We have, first of all, the neuronally expressed genes that are important for the function of motor and sensory neurons and their long axons. And mutations and variants in these genes lead to axonal CMT types, also known as CMT type 2. These genes are typically genes involved in RNA processing, in protein synthesis and trafficking, in the protein degradation, in axonal cytoskeleton, but also ion channels that have to do with excitability or regulation of the blood, brain, and blood spinal cord barrier. Then we have the important genes that are expressed in myelinating cells, the Schwann cells, and they are typically components of myelin itself. The most common genes are actually myelin proteins, PMP22 and myelin protein 0 in the compact myelin and connexin 32 in the non-compact myelin, but also other genes involved in mRNA processing, mitochondrial function, endosomal sorting, and cell signaling. And variants in these Schwann cell genes lead typically to demyelinating CMT type, CMT1, and CMT4. So where do we stand today in terms of therapy developments for CMT neuropathies? We all know that there is a long pathway from the discovery of the genetic cause of the disease with the initial experimental work in vitro, then in small animal models in vivo, and then testing in large animals before making the big step to the human trials from phase 1 to phase 4, and finally to regulatory approval. So it's a long process. If we look at the current therapies for CMT neuropathies, we can see that drug therapy development is clearly more advanced. So there are many small molecules that have been already tested or are being tested at the preclinical stage that are nearing clinical translation that hold promise for having therapeutic effects in various CMT types. Some of them are type specific and some of them are horizontal and may be beneficial for multiple CMT types. And I will go into detail for some of them. And then if we want to address the genetic cause of the disease, then we need gene therapies. And in terms of gene therapy development, we're lagging a little bit behind, with most studies still at the preclinical level, either gene silencing or gene replacement approaches, but some have already entered the clinical stage. We have a clinical trial recently started for CMT1A with a microRNA gene silencing, and we also have a gene replacement trial for CMT2S. So I will show you some examples of this. But in essence, we do not have any EMA FDA-approved therapy specifically for CMT neuropathies yet. So still a long way to go, but we have made, hopefully, I will be able to convince you a lot of progress over the last decade. I will start first with the pharmacological therapies for CMT neuropathies. And some of them are already in clinical trials. I borrow this nice diagram from the De Grado et al. Review about various treatments. And you can see that various small molecules target different pathomechanisms in CMT neuropathies, either involving myelination or axonal degeneration or other complications such as inflammation. Some of them target specific types because of the unique pathogenesis of these types. And the first prime example is the sorbitol dehydrogenase neuropathy, where aldose reductase inhibitors are used to reduce toxic sorbitol levels. Then we have another trial that is quite unique, the L-serine trial for the hereditary sensory neuropathy type 1. Which is currently ongoing. And then we have non-specific targeting, for example, with a chloride 1 muscle channel that is tested by NMD Pharma to enhance neuromuscular junction function. Then we have several small molecules that are at the preclinical stage. Actually, some of them already entered clinical trials recently. First of all, the HDAC6 inhibitors. The clinical trial phase 1 started earlier last year. And then we have Cefin, which is targeting the endoplasmic reticulum stress and unfolded protein response. We have the neuregulin pathways that target mostly myelin thickness regulation in selected CMT types. We have other approaches that target inflammation, which occurs quite prominently in some CMT types. We have SARM1 inhibitors that block axon degeneration, and very specific approaches, for example, for the TRPV4 channel inhibitors in CMT2C. And finally, the GCN2 inhibitors for the tRNA synthetase associated CMTs. I will give you some examples of these in the following slides. I will start with probably the leading therapeutic currently in clinical trial that has completed already phase 2-3 testing. This is about the sorbitol dehydrogenase neuropathy or SORT-CMT. It's a relatively recent addition in the CMT family of disorders. discovered by Andrea Cortese and Stefan Sugner in 2020. This disease is autosomal recessive, is an axonal type of predominantly motor neuropathy caused by biallelic mutations in the sorbitol dehydrogenase gene that is responsible for metabolizing sorbitol into fructose in various tissues and particularly in the axons. So when we have a loss of the SORD function in SORD CMT, we lose the ability to metabolize sorbitol. There is accumulation of sorbitol up to 100 times the normal levels in axons, and that causes various toxic effects, especially to neurons, mitochondrial dysfunction, and so on, leading gradually to axonal degeneration and the manifestations of neuropathy. Luckily, this sorbitol elevation can also be used as a biomarker because it is found to be elevated in blood. And so the approach here is to actually stop the production of the toxic sorbitol by going one step upstream and blocking the previous enzyme, which is the aldose reductase. So aldose reductase inhibition will reduce the production of sorbitol and ameliorate the effects of SORD deficiency. So this approach has been demonstrated to be effective initially in patient fibroblast. It showed significant reduction of the SORD levels in vitro in the fibroblast. And then in the Drosophila model of the SORD neuropathy, it also resulted in functional improvements, both in the mobility and also in the neurodegenerative readouts. So clearly there is a target engagement to reduce the toxic sorbitol levels with this approach. This treatment then also was tested in an initial clinical trial, pilot testing in 8 patients. And there it was demonstrated that up to 66% mean reduction of sorbitol levels can be achieved with aldose reductase inhibitor. And this then finally led to the phase 2/3 study, INSPIRE, which was a double-blind, randomized, placebo-controlled, multi-center study including 56 SORD patients older than 16 years old. This was a 2-to-1 randomization, and the result of this INSPIRE study was that there was consistent, significant lowering of blood sorbitol levels. There was also a correlation of reducing sorbitol level and functional outcomes such as the 10-meter walk and run test. And there was also improvement in the lower limb function in the CMT functional outcome measure. However, the primary endpoint, which was the 10-meter walk run test, was not statistically improved. Interestingly, another patient-reported outcome, the CMT Health Index, showed significant response to treatment. This study has now moved to an open label extension, and recently data from 18 and 24 months were presented indicating sustained results as seen with 12 months of treatment. And also importantly, one of the important imaging biomarkers, the intramuscular fat accumulation measured by MRI that was presented by Professor Riley last week, showed significant improvement after treatment with the govorestat. So we hope that this approach will proceed for further development and towards regulatory approval. I will switch now to the SARM1 targeting, which has been discussed for several years now. The interest in SARM1 inhibition started with some initial evidence from chemotherapy-induced and diabetic neuropathy, where inhibition of SARM1 can improve the outcome. SARM1 is the primary regulator of axon autodestruction. So it's an executor of axonal degeneration and seems to be playing an important role in the final common pathway of multiple pathologies leading to axonal degeneration and is related to the imbalance between NAD and NAM levels. In the CMT space, it was first shown in the CMT2A rat model to result in significant rescue of the phenotype. And so that created a lot of interest. However, subsequent testing in other CMT type models, including the CMT1A, which is a demyelinating type, and another three axonal CMT models, the CMT2D, 2E, and 2S, unfortunately failed to show any benefit the inhibition of SARM1. So SARM1 inhibition is unlikely to be a magic bullet for many CMTs, but maybe a promising approach, at least for the CMT2A. Clearly, we have learned from these recent findings that we need to identify biomarkers that will be an indicator for whether SARM1 activation is indeed playing a role and whether inhibition will be beneficial for a particular CMT type. Another very promising avenue in pharmacological therapies is the histone deacetylase 6, HDAC6 inhibition. Histone deacetylase is an important molecule in axons of peripheral nerves. HDAC6 deacetylates alpha tubulin, which is a component of the microtubule cytoskeleton, crucial molecule in axonal transport. So there is degradation of toxic misfolded proteins as a result of reducing HDAC6 function. There is also a detrimental effect of HDAC6 on axonal transport because of the destabilization of microtubules and impaired binding of the motor proteins, and this can be reversed by inhibition. And finally, at the axon terminal, there is also promotion of axonal degeneration by HDAC6 through the F-actin network, and this can also be reversed. So HDAC6 inhibitors showed promise in models initially of Hitchcock protein B1 neuropathy, the CMT2F. So HDAC6 inhibitors resulted in functional improvement in this model, particularly the axonal deficits. And then in other models, including CMT2A and 2D, as well as recently in CMT1A, it was shown that HDAC6 inhibitors can have beneficial effects and improve functional outcomes, or even reduce the aggregations of PMP22. HDAC6 inhibitors have now been taken to the first in human phase 1 clinical trial by Agustin Therapeutics. The first patient has been dosed in May of last year. It is considered to be a medication that can be used for long term in such a chronic disease. It is orally bioavailable. It has shown not to have any off-target effects. Relatively safety profile was good, and also had a good penetrance to the peripheral and central peripheral but not central nervous system. So we are excited to see the further results of this ongoing clinical trial. Another therapeutic target that has been developed by NMD Pharma is the inhibition of the muscle chloride channel. Muscle chloride channels play important role in the stabilization of the muscle membrane, and that can have an effect on the neuromuscular transmission and muscle excitability. So it was the whole idea here started with the finding that if you do electrophysiological testing of neuromuscular junction in CMT patients, you will see abnormal neuromuscular transmission as shown by the single fiber EMG jitter, increased jitter. And there is also jitter and blocking as we see with neuromuscular junction disorders, both in CMT1 and in CMT2 patients. So that seems to be a more general aspect of CMT pathology. So testing of the chloride channel inhibition in models of CMT1A and CMT2D resulted in significant functional improvement, although it was a short-term evaluation in this model, but at least in the short term, there was a significant functional benefit. So in my knowledge, NMD Pharma now is planning to perform a longer clinical trial also in patients. So that may be an add-on or a symptomatic treatment that may enhance neuromuscular transmission in various CMT types, regardless of the cause. A particular CMT type that has been also very intriguing is the CMT2C. It's an axonal type that is caused by variants in the TRPV4 channel. This is a channel that plays important roles in excitability. It's permeable to calcium and causes a progressive phenotype with predominantly motor neuropathy with variable severity. And further functional studies in the variants that cause CMT2C indicated that the variants result in a gain of function. So there is prolonged opening of the channel, resulting in toxicity and nerve pathology. Further work that has been published a couple of years ago by Charlotte Sumner's group, Sullivan et al. Showed that TRPV4 mutations lead to toxic gain of function. There is increased opening of the channel and increased calcium influx, because if you knock in the variants that will cause the disease, but deletion of the channel will not cause the same pathology. It actually will rescue the pathology if you remove the channel. Further work showed that probably one of the major pathological mechanisms is the breakdown of the blood-spinal cord barrier and leakage into the spinal cord that indirectly causes toxicity to the neurons, the motor neurons. So the approach here that is now being tested for treatment is to inhibit this TRPV4 channel with a specific antagonist, the GSK219. This has been tested in the mouse model, the knock-in mouse model of CMT2C, showing a beneficial effect. It showed that the breakdown of the blood-spinal cord barrier can be reversed, and also the functional benefits were demonstrated in this model. Currently, this treatment is being developed for clinical testing by Actio Bioscience. So a phase 1 trial is in progress, enrolling initially healthy volunteers. So that may be another very specific treatment for this particular CMT type. Another interesting group of CMT neuropathies that may be amenable to pharmacological but also gene therapy treatment are the ones associated with tRNA synthetase variants. There are 6 different types. Of course, the most common CMT2D, that is caused by variants in the glycyl tRNA synthetase or GARS. These synthetases are responsible for loading the amino acid to the tRNA in order to enable the translation of the proteins in the ribosomes. If you have a variant, pathogenic variant in the tRNA synthetase, then that will cause impairment of release of the tRNA from the synthetase to the elongation factor, and that will cause a blockage of the protein translation. At the same time, there is a depletion of the tRNA, and this is work done by Rob Burgess and Eric Storkenbaum labs over several years. This pathology will trigger an integrated stress response and GCN2 dependent activation. So the GCN2 seems to be an important regulator of this abnormal integrated stress response that occurs in neurons. So based on this, there was an effort to target GCN2 therapeutically. First of all, it was shown indeed that it's upregulated along with the integrated stress response in motor neurons in the model of CMT2D. And then either genetically deleting GCN2 or pharmacological inhibition of GCN2 resulted in a therapeutic benefit in the model of CMT2D. So this opens the way for small molecule targeting of the GCN2 for therapeutic targeting. I will then now switch to genetic therapies for CMT neuropathies. And when we discuss genetic therapies, we have to consider the actual molecular genetic cause of the disease because we need to be specific in terms of how we intervene at the level of the genetic defect. So for neuropathies that are dominant, so either because of an excessive gene dosage effect, as in CMT1A, or by dominant variants in genes like myelin protein 0, we need to silence the gene. So we need the gene silencing approach. For autosomal dominant mutations in myelin protein 0, we may need allele-specific silencing, editing, or knockdown and replace approaches. Other neuropathies result from a loss of function. I will show you two examples of that, CMT1X and 4C. And in those cases, we need to replace the healthy gene in the affected cell type. For all demyelinating CMTs, we need to target Schwann cells because this is where the defect leads to the pathology. For the axonal CMTs represented by CMT2A and mitofusin 2 mutations, these are also autosomal dominant. They cause toxic effects in axons, and here we need to enhance the normal protein or knock down and replace or use allele-specific silencing. There are also some non-specific approaches, and I will show you an example of that as well. So I will start with targeting neurons for gene therapies in axonal CMT type 2 neuropathies. So targeting neurons is a relatively easier undertaking because there is already an established approach for gene delivery to neurons. This is basically the result of a lot of treatment development for spinal muscular atrophy, where motor neurons are targeted for gene replacement. Here we can use intrathecal lumbar injection. Intravenous delivery will require much higher doses because of the blood-brain barrier, or we need viral vectors that can penetrate the blood-brain barrier also beyond infancy. Several viral vectors such as AV9 and AVRH10 have shown the ability to transverse the blood-brain barrier at least up to a certain age, and also to have a tropism for neurons. Of course, there is an ongoing effort to find new viral vectors and also non-viral vehicles to deliver genes to neurons. Two of the most promising approaches is the AAV-PHPN, which has shown significant neuron specificity in preclinical work. And also, currently, there is a lot of excitement about the transferrin receptor targeting with the adeno-associated viral capsids because the transferrin receptor is located on the blood-brain barrier and can facilitate penetration of viral capsid into the CNS and to the cerebrospinal fluid compartment. One of the gene therapies that has been promoted for CMT2D is using the BDNF trophic factor. So BDNF seems to be involved in the pathogenesis of CMT2D based on the work by Slay et al. Because it interacts with the TrkB and affects the protein synthesis and impairs the axonal signaling also at the neuromuscular junction and the axonal transport. So here the effort is to replace the BDNF, which is depleted in axons, So an intramuscular delivery of adeno-associated viral vector 8 with a muscle-specific promoter to express the BDNF trophic factor, and that is delivered to the axons, have shown in preclinical work that it can reverse the axonal transport defect that the CMT2D animal model has. So this could be an approach that is specifically targeted for CMT2D. There is also the allele-specific silencing for CMT2D. It was tested previously in Rob Burgess' lab. So this was based on delivery of allele-specific microRNA silencing in two different models of CMT2D with variants in the GARS gene. And in both cases, if the treatment is delivered very early in postnatal day 0 to 1 in this model, you can have a significant improvement of pathology and function. There are two caveats in this approach. The first one is that the specific models had a big difference between the mutated and the healthy allele from 5 to 12 base pairs difference. So that was relatively easy to perform allele-specific silencing. This may be more challenging for variants that are only one base substitution. The other important message from this study was that if you delay the treatment, for example, after 1 week of age with intrathecal delivery, the benefit is only partial, but this is a common theme in all treatments. The later you intervene, the lower the therapeutic benefit is likely to be. Finally, the most recent development in CMT2D gene therapy is the tRNA replacement, because as I have shown you before, there is a depletion of tRNA because of the dysfunction of the tRNA synthetase and segregation of the tRNA. There is a loss of tRNA, and that will cause blockage of protein translation. So the idea here is to overexpress transgenically the tRNA. It was initially shown by transgenic overexpression that this can rescue the CMT2D models. And then currently there is an adeno-associated viral vector 9 mediated tRNA-based gene replacement therapy that is being tested by Eric Storkenbaum, and XT-RNA Bio is a company that is leading this gene therapy towards clinical translation. I will also talk very briefly about CMT2A, which is, as you all know, the most common axonal CMT type. It's responsible for about 10% of all CMT neuropathies. It's a severe progressive neuropathy and results from dominant mutations in most cases of the mitofusin 2 gene. Mitofusin 2 is an outer mitochondrial membrane protein that is important for the fusion and mobility of mitochondria. Various studies from Bobello lab and others have shown that mitofusin 2 mutations result in impairment of mitochondrial mobility, impaired energy production, and axon growth deficits. So what can be done to treat CMT2A? There are different approaches. One of these is the work done by Stefania Gorti's lab in Milan. They used a knockdown and replacement approach. So this is a combination of using a silencing RNA molecule to silence the endogenous mitofusin 2 gene, and at the same time with the same expression cassette to express the healthy copy of the mitofusin 2, which is resistant to the silencing shRNA. So using this approach, they showed a therapeutic benefit in patient-derived motor neurons in vitro, but also some promise in vivo in the mouse model of the disease, although functional benefit was difficult to demonstrate because this is not a very robust disease model, but there is further effort to develop this as an approach. A similar approach is also tested for CMT1B. Another approach to treat CMT2A is to overexpress compensatory mitofusin, either mitofusin 1, which is a similar mitofusin but expressed at very low levels in neurons normally. So it was shown initially by Bob Bello's group that if you increase mitofusin 1 in neurons transgenically, you can actually improve the phenotype in the model of 2A. And then in my lab, we used mitofusin 1 overexpression to reverse the aggregation of mitochondria and axonal defects in another model of CMT 2A. So upregulation of mitofusin 1 may be an approach. More recently, a very nice study by Nathalie Bernard-Marisal in Marseille has demonstrated that if you overexpress the wild-type mitofusin 2, this can have a beneficial effect in a model of CMT2A. So here it was AAV9 based delivery of mitofusin 2 wild-type allele under a neuronal promoter, the SIN1 promoter, intrathecal delivery in the mouse model of CMT2A. This resulted in significant targeting of motor neurons and then at the functional level improvement of neuromuscular junction denervation and also motor performance. So this is a very nice study and hopefully will also lead to further development as a treatment approach for CMT2A. There is a study that has been going for a while for treatment of CMT2S. This is an IGH MBP2 loss of function gene associated disease that looks like a motor neuron disease, like a spinal muscular atrophy, but in milder cases, more like an axonal neuropathy. And this has been tested initially in the mouse model of the disease. And currently there is an ongoing clinical trial by Alkion Therapeutics in children 2 months to 14 years old. And we're still waiting to see the results of this. But this is an example of gene replacement in neurons. Another gene replacement approach, mainly targeting neurons, is the CMT4J gene therapy, where we have the FIC4 gene loss of function variants causing a motor neuron and a mixture of demyelinating and axonal neuropathy. So this was tested again in the mouse model of the disease initially and showed a significant therapeutic benefit. Of course, the earlier the treatment, the better the outcome. And this is currently being further developed by Elpida Therapeutics. There is an ongoing natural history study for CMT4J patients that is expected to lead directly into a clinical trial. And this approach has already passed IND approval by FDA. In the last part of the talk, I will talk about gene therapies for demyelinating CMT neuropathies. So as I said at the beginning, to treat demyelinating CMTs with gene therapies, we need to target Schwann cells because this is where the disease starts. So this is a bigger challenge than targeting neurons because Schwann cells are distributed throughout the nerves of our bodies. So we need a wide biodistribution, and this brings also various issues of optimal dosing, optimal delivery, and clinical scalability. So there are different methods to deliver genes for targeting Schwann cells. There is an intrathecal approach that we use in my group. There is also an intravenous approach that is less preferable because it requires very high doses. And there is also an intraneural and more recently perineural approach by the Nicolas Tricot group. And there are also intramuscular approaches for trophic factors. Always we try to find vehicles that will efficiently target Schwann cells. Recently, there is more work to find new capsids, new viral vectors, and there is some promising work, for example, with AAVF and PEP2 SC1 and SC2 that were developed in human Schwann cells. I will start first with CMT1A, which is the most common type of CMT. This is clearly a gene dosage effect related disease. So if you have duplication of PMP22 gene, there is overexpression of PMP22 leading to CMT1A. If you have a deletion, you have lower levels of PMP22 and hereditary neuropathy with pressure pulses. So in order to treat CMT1A, we need to silence to some degree the PMP22 gene. There are non-viral methods, including the Falcon technology, the fatty acid ligand conjugated oligonucleotide. And there is also the squalene nanoparticles approach, delivering siRNA. And we also use vectorized approaches with AAV vectors delivering microRNA to target PMP22. In all cases, we want to achieve a normalization of the PMP22 level around 100% of normal, not excessively silenced and not and still being effectively silenced to reduce the toxic levels. The most promising treatment that is currently actually just entered clinical trial is the fatty acid ligand conjugated siRNA approach, the Falcon technology developed by DTX Pharma. It was tested initially in mouse models of CMT1A, showing therapeutic benefit, improvement of the pathology, improvement of the motor function, and then also in non-human primates. This approach was acquired by Novartis 2 years ago and actually just entered a first-in-human clinical trial. This was initiated in Canada in 2 sites. 4 different doses intravenously are used. We don't know the exact protocol of the frequency and the exact dosing, and this is compared to placebo. But this targets CMT1A patients, a total of 28 individuals in this initial testing. So there will be a lot of excitement to see how this turns out to be. And that will be a very important study also for further development. There is also another non-viral approach with squalene nanoparticles delivering siRNA by the Lilian Massad group in Paris. This showed beneficial effect in the mouse model of the disease and recently was also demonstrated in non-human primates. So we also wait to see how this will be developed towards clinical translation. There is an approach by Nicolas Tricot group with shRNA targeting the PMP22 gene. This was initially tested with direct injection into the nerve, resulting in improved pathology of the injected nerve. And then recently by this group and developed by NervoSave, it was shown that perineural injections, so outside the endoneurium, can actually have a beneficial effect. This, of course, requires multiple injections in multiple nerves, and it's a procedure that requires anesthetic support. But it did show very effective targeting of Schwann cells in the injected nerve and functional improvements. In my lab, we have developed a microRNA gene silencing for CMT1A that we delivered intrathecally with AAV9. This work has shown that we can selectively and effectively silence the PMP22 gene in the peripheral nervous system, improving the phenotype of the model. More recently, we tested that also in a dose escalation study, reproducing the initial results and also improvement of biomarkers. I will not go into detail. We also showed with RNA-scope and dual detection of microRNA and PMP22 that silencing is uniform in Schwann cells within the peripheral nerves and that we don't cause an excessive silencing. This work has also proceeded to scale up testing in non-human primates. We extrapolated the doses based on the CSF volume From the effective doses in the mice, we went to the non-human primates in two different doses. First of all, we showed that there was no toxic effect and no safety issues in primates. And the important finding is that we could actually achieve significant biodistribution in peripheral nerves with both doses at 6 and 12 weeks after injection. And there was also target engagement with silencing of PMP22, demonstrated both by Western blot and by real-time PCR at the protein and mRNA level. And this silencing was in the range of 30 to 50%, which is what we want to have for CMT1A. The second most common type of CMT is CMT1X, is the X-linked form of the disease. It's about 15% of all CMT cases in most populations. And the disease is caused by mutations in the gap junction protein connexin 32, which forms channels of communication between the Schwann cell cytoplasm outside the myelin sheath and the inside of the myelin sheath in the areas of non-compact myelin. These gap junctions are crucial for maintenance of homeostasis and integrity, not only of the Schwann cell itself, but also of the axon. And we have shown in various preclinical studies that the mutations of gap junction 32 lead to a loss of function in the majority of cases. So the strategy here is to replace the connexin 32 gene. This needs to be cell-specific because the expression of connexin 32 is only found in Schwann cells in the peripheral nervous system. So we cloned the connexin 32 gene under the myelin-specific myelin protein 0 promoter and delivered this by lumbar intrathecal injection using the AAV9 vector. And we showed in different models, including the knockout mouse model, but also models that express variants reported in patients, that this treatment can lead to functional improvement, improvement of the pathology, and expression of gap junctions in the non-compact myelin areas of the mouse model. We have also tested an AAV-RH10 vector at different doses, showing similar efficacy as AAV9 in a dose escalation study, and also having no toxic effects. And finally, recently, we completed a trial of scale-up of this CMT1X gene therapy in non-human primates, delivering the connexin 32 gene either with AAV-RH10 or AAV-RH74. We showed the dose-dependent biodistribution, so the higher dose, the more effective targeting of spinal roots and peripheral nerves, and expression specifically in Schwann cells. So we hope that this program can move also towards pre-IND application to FDA. Finally, for CMT4C, which is probably the most common autosomal recessive CMT neuropathy of the demyelinating group. This accounts for 3 to 4% of all non-CMT1A demyelinating neuropathies. It is caused by biallelic mutations in the SH3 TC2 gene, which is important in myelinating Schwann cells for the maintenance of myelin and the development, working in the endocytic recycling compartment. And so here we develop again a gene replacement strategy for SH3-TC2 under the control of a minimal myelin protein 0 promoter. We demonstrated that with this gene therapy, we can achieve therapeutic benefit in the mouse model of CMT4C. And recently we completed the dose escalation study with different doses of the SH3-TC2 therapeutic vector, showing a dose-dependent improvement in biodistribution, expression, and also functional benefits in the mouse model of CMT4C. There was also similar improvement in the pathology, myelin thickness, and the number of demyelinated fibers. And this program has also completed the pre-IND application to FDA and now going through final preclinical testing before moving to the clinical translation. I will close with a summary of the current gene therapy developments. I showed you some examples of gene therapy for axonal CMT types. Some of them are already in the clinic. Some of them are nearing the clinical translation. These therapies target neurons because we have to fix the problems in neurons and axons, and they're mostly delivered by intrathecal injection. Then we have several approaches for demyelinating CMT neuropathies. For the dominant ones, we have to use gene silencing or allele-specific silencing. Prime example, CMT1A with the Falcon technology already in the clinic. And then for the loss of function ones, we have to replace genes again in Schwann cells, and various delivery methods are being evaluated. There are also the non-specific approaches, for example, neurotrophin-3, SARM1 inhibition that I mentioned at the beginning have been tested along with BDNF. So with this, I would like to thank you. I would like to thank the members of my team in the neuroscience department who did all the work I showed you from my lab. I would like to thank many collaborators who worked with us over the years for the results I showed you, and also funding from patient associations and some of the industry partners as well. And I would be happy to hear any questions you may have. And thank you again for your attention. And I will do the moderation also because Davide unfortunately could not join us today. So I will look in the question and answer box, and I see Filippo Genovese. Hi, Filippo. Filippo wrote, thank you, Kleopas, for this comprehensive and optimistic insight of the different research and treatment. Let's not forget, as already mentioned in other webinars of this series, that we do already have some options to feel better, a healthy lifestyle, staying active, and surgery when needed. Of course, thank you very much, Filippo. So I focused here, of course, on pharmacological and gene therapies, more for the future, but in the present, clearly, the most important thing that patients with CMT can do is really, all the rehabilitation measures, the regular physiotherapy, assistive devices as needed, and of course, in specific indications, surgery to improve functional outcomes. So clearly, thank you very much for this point. And therapists, of course, have a major role to provide a benefit for patients until we have effective therapeutics in the clinic. I don't see any other questions in the Q&A. And Elizabeth, I don't know if you see any other ones in the chat.
No, just a thank you from José Luis.
Okay, thank you very much. Yeah, thank you again for joining. Thank you, Elizabeth, for arranging
Thank you,
this.
Kleopas.
I guess if no more questions.
There's a question in the audience.
Yes.
Actually, Ingolf Pernis want to talk. So...
Yes, please, Ingolf.
Yes. Hi, thank you very much, Kleopas. This is extremely helpful and enlightening, although in some parts, of course, difficult for a layman to understand deeply. But my question in this would be, given all these different and so many approaches on therapeutic measures to help in CMT, if you could tell us what are the most important roadblocks? What makes your work difficult, so difficult, and what could patients, perhaps patient organizations, or even political leaders do to take away these roadblocks, if there's any?
Yeah, thank you very much, Ingolf. And of course, there are many roadblocks. Maybe I indirectly pointed out to some of them, but I'm glad you asked this because this is very important. I haven't mentioned the question of clinical trial readiness, but this was covered by Professor Riley last
Yeah.
week. It's clearly a major roadblock, or it has been in the past, hopefully not so much in the future, but it remains a challenge to be able to demonstrate a response to any treatment that we test in the clinic for this group of very rare and very slowly progressive disorders. So, as you know, if we have a very rare disease, for example, with only 12 or 20 patients participating in the trial, and between them there is a variation in the severity, variation in the way they progress, it will be very difficult to prove that a certain treatment benefits these patients. So we need to have sensitive clinical outcome measures to be able to tell that they really had a benefit from any treatment. So that's a major challenge that many clinicians have been working on to develop more sensitive outcome measures. You've seen the one example with the short neuropathy trial. So some of the outcomes were responsive and some of them failed to show a significant benefit. But this is inherent to this group of disorders because they progress very slowly, which is good for patients, but can be challenged for proving benefit of therapies. Then we need the biomarkers because we need to be able to measure a benefit in a time scale that is reasonable and possible, feasible for the industry to support financially. So if a company cannot demonstrate an effect of their medication or gene therapy within a year or a year and a half, that may not be financially feasible. If they had to, run the trials much longer, then they don't have enough resources to do that.
Yeah.
So I think that just shows the limitation. And of course, many of the companies in this space have their own challenges. Some of the companies have invested in some stages, then they have their own financial challenges and they were no longer able. There is a lot of risk in the pharmaceutical industry. Some of them are startups. There is no consistent industry funding. The only example that we have from a big pharma supporting CMT therapy is the Novartis trial, as far as I know, that a big pharma really is now taking a treatment for CMT neuropathies in the clinic. All other clinical trials that I described are mainly from small companies that have their own, instability and within the financial and regulatory environment. And of course, we need the patient associations because they can advocate to the industry, they can advocate to the regulatory authorities, they can help us and guide us to choose the right outcome measures, what really matters to patients. That's why we more and more incorporate patient-reported outcomes in our studies to help us develop treatments, in a more successful way than what we have done in the past. So, patients and patient associations are partners. There is also lack of funding for preclinical development many times. So there are ideas that may not be able to be, further developed because there isn't enough funding for such rare diseases. Europe has other priorities for more common diseases, perhaps not health-related funding. So it becomes challenging to really support, consistent funding for researchers working in CMT neuropathies. I hope that was
Thank you. Yes.
somewhat, a response to your points, but of course there is a lot of discussion that we can still do. I see one more comment here. So that's from Rida Boulos. Thank you for the insightful conference. So Rida is a Lebanese neurologist. We know humans can have pre-existing neutralizing antibodies. Yes. Um, which poses problems of redosing, and also AAV can only accommodate around 4 kb of DNA. For example, for C, yes, exactly. Seeing these limitations, do you see a future role for autologous gene editing, for example, in 4C to mediate the correction of the gene followed by the reinfusion of cells? Okay, there are many points here. I think the first point about the pre-existing immunity is something, of course, very important. If there is pre-existing immunity, certain amount of neutralizing antibodies that patients may have could actually cancel the effect of gene therapies because they will neutralize the virus, and also they can generate immune responses that can be toxic to the patient. So definitely screening for pre-existing antibodies should be part of any clinical protocol. For all gene therapy protocols, there may be also the need for immunosuppressive um, treatment together with the gene therapy to block any possible immune responses that can be toxic. We're definitely learning a lot from other diseases that have already clinical stage therapeutics like Duchenne muscular dystrophy, like spinal muscular atrophy. So experience from these gene therapy trials will definitely help us as we move to the clinic for CMT neuropathies. In terms of the size, yeah, indeed, there is a capacity limitation for the AAV vector, which is the most commonly used. For the CMT4C, we just stayed below the limit by using the minimal promoter, but some other genes may not be possible to package in AAV. In my group and some other groups, we're working on the development also of non-viral vehicles, for example, nanoparticles that may carry bigger genes, but they have their own limitations and they're still at very early stages. And there is, of course, the option of gene editing within the genome. As you know, that has been tested for TTR amyloidotic neuropathy, but that targets the liver. If we need to target these treatments to Schwann cell, the efficacy in vivo may not be what we expect, at least at this stage. I think there is still more technological development to be done before we can have efficient in vivo gene editing. There are also mRNA gene editing approaches. So there is a lot of development, and hopefully in the near future we will have more tools in our possession to be able to, maybe propose more sophisticated approaches for some of the CMT neuropathies. Okay, thank you very much. I don't see any other question, and
There's no more
we already
question.
passed the hour. Yes. So thank you again for joining today, and thank you for the opportunity. And
Thank you, Kleopas, for your
thank you.
lecture and the webinar series. Oh, I think someone, oh, no, it was a mistake.
Okay.
Thank you, Kleopas. See
Thank
you
you
next
very much.
time.
Yeah. Have a nice
Bye.
evening. Bye bye.